All right, so I'm going to talk about inhibitory control a little bit, you know, just to introduce you to what that is. Inhibitory control, obviously, being a control process that allows us to implement certain behaviors like action stopping. So if you're about to step into the street here, and you see a car bearing down, you can stop yourself from finishing and executing that movement in time using inhibitory control. Now in the lab, obviously, we don't test this by shoving people in front of moving cars. We test it using the stop signal task, which is what Berta just so eloquently explained. So I'm not going to go into too much detail. You get a go signal on every trial that tells you left or right in this case. And then on a subset of trials, you'll get a second signal, the stop signal, that tells you to try to stop that action. And you can play around with the delay, like Berta said, and make it easier or harder for people to stop themselves. Now, this task comes with a standard cognitive model, which is goes back to Gordon Logan's seminal work. And basically, the supposition here is that the go signal triggers a go process that works towards executing a movement, and the stop process executes the stop signal starts a stop process that works towards or races the go process. And they both race to a threshold, and whichever one finishes first on any given stop trial determines whether you're able to successfully intercept and stop the response. And there's some adaptations to this model, and there's some alternative views that deal with whether these processes interact and how and whatnot. But generally speaking, this model is still the standard accepted model in the field, whether you buy into the adaptations or not. There's also what I would consider, at least in humans, to be a standard model of action stopping in humans. So you have this ostensible right lateralized three-pronged network that includes the pre-SMA, the right IFC, and the subthalamic nucleus. And the idea here is that the the cortical regions signal down to the subthalamic nucleus to kind of stop these thalamocortical motor loops that are underlying the action. Now, just as with the race model, there's challenges to this model as well. Some of them come from the non-human animal domain, often in tasks that involve eye movements and things like that. Some of them have to do with Berta's work, and we can talk about those. But the one criticism that I want to talk about in the purely in the human domain today is Adam Hampshire's great work, which basically uses uses tasks like this. These tasks have been used in the literature many, many times. It's basically a stop signal task that includes an additional infrequent event that is not paired with a stopping instruction. So basically, you're just doing a standard stop signal task, left, right, and then an up arrow means, in this particular context, a stop signal, but then the subjects also know to expect a down arrow, which they're supposed to ignore or do something else. Typically, the instruction is to ignore them. And so Hampshire and colleagues and some other people have found that these infrequent stimuli that are not paired with a stopping instruction activate the exact same regions as the stop signal, including in the right inferior frontal cortex. And that's kind of the big take-home message. And in fact, in their abstract, they say, well, we propose based on this that there aren't any inhibitory modules within the frontal lobes at all. So that's a big challenge to this theory. And that's the first problem in the literature that I want to address with some empirical data, and later on a little bit of a theoretical conceptualization first, which is how can we disentangle attention from inhibition, not just in the stop signal task, but much more generally speaking. And that's probably not a question. So attentional capture obviously being something that happens on infrequent events, whether they're being paired with a stop signal or not. And this is probably not a question that we can answer with fMRI, just given its time resolution. I mean, the go and the stop process occur very, very closely together in time, and even more so, attentional detection and inhibitory control as part of the joint stop process, are very, very closely entangled in time as well, and occur right sequentially after one another. So what we probably want to use to answer this particular question is a method that has time resolution, that has the potential to disentangle these processes. And that brings me to neurophysiology and some of our work. And so this is just a very simple ERP study. And it basically goes back to the proposition of De Jong and Creton and others in the 90s, that this frontocentral P3 waveform in the EEG may be related or may actually index the inhibitory control process in the stop signal task. And in this study, we found some really nice properties of this frontocentral P3 that speak in favor of this potential interpretation. First of all, this frontocentral P3 starts earlier on successful versus failed stop trials within subjects, and that maps nicely onto the predictions of the race model, because if the stopping process, or at least the ostensible stopping process, starts earlier, then you would be more likely to succeed at stopping. So that makes sense. And moreover, the onset of this frontocentral P3 is also very highly correlated across subjects with stop signal reaction time, which is a behavioral measure that you can extract from the stop signal task when you're applying the assumptions of Gordon-Logan's race model. And so what these data here show is that subjects with a faster stop signal reaction time, i.e. a faster stopping process, also show an earlier onset of this frontocentral P3. So this paper is, accepted somewhat, it gets cited and whatnot, but there's also a lot of criticism of this supposition that ostensibly this frontocentral P3, reflects inhibitory control. And one of the chief criticisms comes from my good friend René Huster, and this is a great study from Lisa Raud and René, who I believe is here. And so what they basically do here is they have recorded EMG from the muscle that you're responding with. And the blue lines, disregard those, it's just the go response, but it's from the unstopped hand. But what's important here is the red line, which is the hand you're supposed to stop. And what they find is that on successful stop trials where you didn't make an overt response, there is still some residual activity in the EMG that you can observe. And importantly, you can see when that residual activity gets reduced, right? And that's ostensibly the first sign of inhibition, or is a overt signature of inhibition on the level of the motor cortex. So when I was talking to René about this was, SFN a couple of years ago. He was staying in my apartment in San Diego, and was like, well, we have this finding here with the EMG. See how this peak happens much, much earlier than this frontocentral P3? Like if we have signatures of inhibition at the level of the motor cortex, where on successful stop trials, the go response becomes suppressed at around 150 or so milliseconds following a stop signal, then how can the frontocentral P3 reflect motor inhibition when it happens later than that? And that's a very, very fair criticism, and I'm going to try to address that in parts, at least today. But first, I want to show you that there's a third index of the neurophysiology of motor inhibition, which again speaks in favor of some of what René is talking about there. And that is measuring corticospinal excitability from the motor effectors of interest. So that here you can use transcranial magnetic stimulation. You can stimulate a specific part of motor cortex and record EMG from the muscle that part of motor cortex controls. And what you will do then is you will produce a deflection in the EMG called the motor evoked potential. And the amplitude of that deflection is a proxy for the net corticospinal excitability of that entire motor tract. And so when you measure this during the stop signal task, you will find that when people successfully stop, the motor excitability of their motor effector that they're stopping is significantly suppressed. But you will find the exact same thing, and that is a very interesting property of this stopping mechanism, that it actually is effector non-specific. So in other words, if you're stopping a motor effector, like the eyes, the motor evoked potential, or the corticospinal excitability from the task unrelated hand, for example, will also be suppressed. So that's the successful stop trials here. And we found this over and over again, us and others, with different combinations of motor effectors. And the reason I'm bringing this up is because it's a third signature that's important, and that I'll talk about here in the new empirical part of this talk. But also, if you look at this with some time resolution, this suppression of the corticospinal excitability, you can see that indeed it starts pretty early on. So this is corticospinal excitability following a stop signal, time lock to the stop signal. And you can see that there is significant suppression already at 140 milliseconds following the stop signal, much, much before the onset of the frontocentral P3. And I should say this is also work that has now been published, or work like this finding has been published in a very nice, very comprehensive study from Jana and colleagues. So this is not just like unreviewed stuff. This is in the literature. Anyway, so that brings me to the second problem, which is the timing of inhibitory activity, where the question is, why do we have these purported signatures of motor inhibition at different times following the stop signal? How does that make sense? Okay, so let me try to solve some of these problems. I mean, solve is a strong word, but I'm going to put our best foot forward, and then we can see where that leads. So the first foray to address some of these issues happened shortly before the pandemic. Carly Iaculo is an undergraduate in my lab, and Darcy, who's a graduate student who you'll hear from later today, ran an experiment that was basically getting close to this Hampshire logic of like just presenting people with an infrequent event that's not paired with a stopping instruction. And the question here is, these subjects never did a stop signal task. They don't know of the stop signal task. They're just doing a motor task where they're making a motor response, and after the go stimulus, they get an infrequent sound that they're either expecting because they practiced with it and they know that they're infrequent, but they can occur, or it's completely unexpected. And that's something that we've done before. But what we found here is that we indeed find a significant reduction of corticospinal excitability even after infrequent events that you're completely expecting but are not paired with a stopping instruction at all. Okay? And so that was kind of the first piece of evidence, but that led us over to a much more comprehensive study, which I'll focus on for the vast majority of the remainder of today's talk here. And this is a study by Josh Tatz, who's a postdoc in my lab, and Shoal So, who's a graduate student. And they basically ran what, a paradigm that's essentially this Hampshire type paradigm, which again has been used by other people in the literature as well, where you have a stop signal task. So you have an instruction that you will sometimes see a stop signal that tells you to cancel the go response. But you are also instructed that sometimes you will see another infrequent signal that you're supposed to ignore and just continue with your response as if nothing happened. Okay? And so Josh and Shoal really ran people through the gamut here. Across several experiments, they basically went ahead and measured all three signatures, neurophysiological signatures of inhibitory control or purported inhibitory control signatures in humans that we know of. And the hypotheses were as follows, and these are pre-registered hypotheses. The first hypothesis was that both stop signals and infrequent events that are not paired with a stopping instruction, which I will call ignore signals from now on, will produce these early latency EMG reductions that Lisa and Renee have found in their study. And I will tell you why we thought that, because we have a whole theoretical framework that Darcy and I have been developing and that I'll talk about towards the end of this talk. Also, we predicted that stop and ignore signals will produce early latency MEP reductions based on the Iaculo study, but we also hypothesized that the amplitude of this MEP reduction will not differ between stop and infrequent ignore trials. And then finally, we used multivariate pattern analysis of the EEG data to really investigate at what point, when you take the whole scalp into account, can you actually distinguish between stop and ignore trials? When does something happen that is unique to action stopping and not just due to the presentation of an infrequent event, whether that's paired with stopping or not? Okay, so let's jump right in here. These are the EMG data. So this is basically a replication of Lisa and Renee's findings. So we have the responding hand here. We have the EMG on a go trial. You can see very nice go-related activity in the motor effector. But you can also see that on successful stop trials, you see this you see residual EMG upregulation or increase. That is then downregulated or dampened or inhibited, following a stop signal. And that happens actually round about 140, 150 milliseconds following the stop signal. And that's exactly what we expected. However, let's bring in the ignore trials. These are the ignore trials. So what we see on these ignore trials is they ramp up and then they ramp back down. And as a matter of fact, they ramp back down with the exact same latency as the successful stop trials. If anything, this decrease in EMG happens a little earlier on ignore compared to stop trials. So it's not significant. So for the purposes of our interpretation, we're basically proposing that this signature happens on ignore trials just as it happens on successful stop trials, and therefore is not a unique signature of action stopping. It does contribute to action stopping, and I can get to that at the end. Let's move on from here to the corticospinal excitability data. First of all, this is again a replication of our expected finding. When you compare go to stop trials, you see a reduction of corticospinal excitability even in task unrelated effectors when there is a stop signal. And now again, let's bring in the ignore trials. And you can see that at least at the very early time point, at this 150 millisecond following the stop or ignore signal time point, you see really equal amounts of corticospinal excitability suppression on ignore and on stop trials. The last part of this is the EEG data, or the EEG data, sorry. There's a lot going on in this figure here, so I'll walk you through this here real quick. So the solid lines here on the bottom is the frontocentral ERP. Here we're looking for the frontocentral N2P3 complex. And what we're seeing is, first of all, in green are the successful stop trials, in red are the failed stop trials. We see earlier onsets for successful versus failed stop trials, as we would expect. We see a P3 on the ignore trials, but it's significantly reduced in amplitude. And we see an N2, but the N2 is actually increased on ignore trials versus successful stop and failed stop trials. And more importantly, up here, you can see the decoding traces that take into account the whole scalp data. And what we basically found here is that while ignore and stop trials start being decodable really early on from go trials or from go signals, the difference between successful stop trials and ignore trials doesn't really emerge until around 180 milliseconds following the stop signal. And moreover, the decoding weights basically tell you that that's because of the frontocentral electrodes, where the ignore trials show greater activity compared to successful stop trials early on. And the first time point where the decoder basically tells us, yes, there is a difference between, there is something uniquely different between stop trials and ignore trials, and the stop trials show greater activity than the ignore trials, is again the frontocentral P3. So let me give you an interim summary here of the empirical part of this talk. How does this address the two problems that I outlined? So the first problem was, why are quote unquote inhibitory brain regions active during mere attentional capture? And that's because attentional capture involves motor inhibition. Anytime you have an infrequent event, you will automatically trigger motor inhibition in the form of reductions of the EMG, reductions of corticospinal excitability, and early EEG signatures that distinguish ignore and stop trials from go trials. And then the second problem is, why do signatures of inhibition occur at two different latencies following the stop signal? And that's because we propose early latency signatures are due to attentional capture effects, and only later signatures are unique to action stopping. So what does that mean in terms of theory? Let's try to pack this into a comprehensive theoretical framework that explains what we believe is going on. And when I say theory, I'm referring to a paper that Darcy and I have just submitted, which is very comprehensive and tries to put all of this into a theoretical framework. But it's basically just an adaptation of Schmidt and Berke's work in rodents, where they proposed what they call the pause then cancel model of the stop signal task. And basically what they find, and this goes to Berta's point, is that early activity after stop signals in the subthalamic nucleus in these rodents that are doing the stop signal task does not differ and does not differentiate successful versus failed stop trials. And only later activity in the striatum, for example, distinguishes between the two trial types, where the ongoing innervation of the go response through the striatum is canceled after the initial, what they call pause, that takes place in the subthalamic nucleus. So the idea here is that stopping is really stopping in the stop signal task is really a two-step process, which basically begins with this very fast, low latency, quote unquote, pause response that is mediated by hyperdirect pathways through the basal ganglia to the subthalamic nucleus that basically shut off the output gate of the basal ganglia. And then you have a slower cancel response, which according to their conceptualization uses archipalatal cells in the globus pallidus. In humans, there would be an equivalent to that. But the idea is that that indirect pathway through the basal ganglia then shuts off the still active innervation of the go response through the striatum. So Darcy and I have adapted this model to humans and to a broader model of not just the stop signal task, but inhibitory control in general, according to which there is a neuroanatomical network underpinning the pause process, where the right inferior frontal cortex is activated by any infrequent event, not just stop signals, a surprising event, a non-surprising event, an infrequent event. In Berke's and Schmidt and Berke's conceptualizations, that includes go signals even. And all salient or infrequent events that produce attentional capture will activate the circuit and pause the go response via the hypodirect pathway from the cortex into the subthalamic nucleus. And then the cancel process kicks in from this arrow here is wrong, from the pre-SMA, and actually cancels the ongoing drive towards the go response in the striatum. And so that basically explains why there would be two different latencies for different signatures of inhibitory control, neurophysiologically speaking, because the idea is that the EMG reduction and the CSE reduction that you find at the level of the motor cortex is actually a signature of the pause process and universal to all infrequent events. And only later processes like the frontocentral P3 perhaps reflect this cancel process, which we actually believe to be, just a specific case of reweighing and reevaluating motor programs that in the context of the stop signal task means cancel, but in the context of other infrequent events that are not paired with stopping can mean something else. So anyway, that is my presentation. I want to thank the people that have done the work that I presented in this particular talk and the rest of the members of my lab and also our funding sources. Thank you very much.